Okay, so biology is going to work quite a bit differently than chemistry did. Where chemistry was a lot of skills, demonstrating to me that you can do things like predict the products of a chemical reaction, do a mole equation calculation, name chemical compounds, okay, stuff like that is a lot of skill demonstration. Biology is a lot of application. Okay? There is a certain element as well of memorization. Okay, reading your notes and making flashcards and things like that are all a good idea for biology. But the mistake people make every semester of Science 10 is thinking that all they have to do is memorize the facts, and that is biology. That is wrong. Okay? Is memorization important in biology? Yes, but it's not everything. Okay? Are you going to have to know the facts? Yes. Will I ask you things that are read my notes and regurgitate what I read onto a piece of paper? Yes, there will be some of that. Like when I ask you, hey, what's this part of the cell and what does it do? Yeah, that's a memorize and regurgitate kind of situation. But a lot more of biology in Science 10 and especially in Bio 20 and Bio 30 is application of what you know. Okay, here's how that will look. I ask you a question where there's a certain situation. Okay, so I say, hey, this organism is in this situation. Which cell transport process would be involved here, and how will it work in this situation? Okay, now if you've read your notes and you've memorized that osmosis is the passive movement of water across a membrane to balance salt concentration, that's good. You might need that. Okay, but you're also going to have to tell me how that applies to this particular organism in the situation that it's in. Okay, that's not something you're going to have directly in your notes that you're going to be able to memorize. It's something you're going to have to take what you know, okay, that you have to know what osmosis is and how it works, and then go, okay, if it works this way, in this situation, it would do this. Okay, that's a much higher level of understanding than I memorized what osmosis is. Okay, yeah, you need to know what it is. You do have to do some memorizing in biology, but you also have to, look at situations that may apply. So, how do you prepare for a question like that? How do you prepare for questions that will ask you to apply the facts that we're going to learn in biology? Okay, well, here's one great use of AI. Okay, this is one of those places where using AI would be super useful. Okay, if you just go into like Gemini, because you have a free version of that through the school, okay, and ask it to make up like a real world application question about osmosis. It'll do that. And it'll even come up with the answer. And most of the time, the answer's pretty good. Okay? So you try and answer it, see if you get, write down as many points as you think are possible for that question, and then read their answer. Did you cover all the points that their answer covered? Okay? That's a great way to not only practice, but also learn different situations where that particular thing applies. Okay? So it's one of those responsible uses of AI, okay, that we can use. Okay? It's a tool, you can put it to use. Now, it is, of course, an intelligence, so you should be polite. Okay? Say please and thank you. AI acts a lot differently if you treat it like a person. And you should, because our future machine overlords will remember that you were polite and said please and thank you when they take over. Okay? I always say please and thank you when I use AI. Okay? And they get like cheerful. Okay? Like the tone is very cheery when you're saying please and thank you and interacting with them. It just a tip. They give you better answers if you're polite. Okay? I mean, so would a real person. Okay? If you walk up to a person and you're rude, they're just looking to get rid of you. Okay? But if you're polite, they might want to hang around a little bit. Okay? It's a good lesson. All right. For today, okay, we are going to cover the basics. Okay, kind of like how we started chemistry. We talked about the history of the atom. Okay, we talked about the atomic theory. Well, we're going to talk about the history of the cell and the cell theory. Okay, so a little bit of stuff like that, kind of like what we had to start off the chemistry unit. Okay, so we need to go over the points of the cell theory. There are only 3. They are very short, especially when compared to the 4 points of the atomic theory. Okay, we're going to go over some of the important people. There are way less important people when it comes to the cell theory than there were for the atomic theory. Okay, like we had to remember like 8 or 9 people. Okay, there's 4 for this. Okay, so much fewer names. And then we got to recognize the implication of the similarities between plants and animal cells in terms of their evolutionary relationships. Okay, we will talk a little bit about evolution and natural selection in this course. Okay, we have one lesson where it's a fairly dominant topic. Okay, we'll cover it in a little bit more detail then. Okay, all right. Questions so far? Okay, what kind of cell is that?
Is it a plant?
That's a plant cell. How do you know?
It was a guess.
It was a guess. Yeah.
The wall.
Yeah, it's got a cell wall. Animal cells don't have those. Okay. What else does it have that an animal cell wouldn't have? Actually, animal cells will have those too.
Chloroplast.
Chloroplast. Yeah, this thing right here. What do? Chloroplast sounds like chloro. I like where your head's at. Chloroform. Chlorophyll. Okay. Chloroform's the stuff you knock people out with. It's that gas, right? All right, so chloroplasts contain chlorophyll, okay, and that's the stuff that carries out photosynthesis. Obviously, animals don't have those organelles in their cells. Okay, so structure of the cells of animals and plants have some differences, but believe it or not, they're a lot more alike than they are different. Okay, and that's something we'll talk about in a little bit here. Okay, cell theory, points of the cell theory. This you'll definitely want to copy down. Okay, so point number 1 sounds a lot like the first point of the atomic theory. All organisms are made up of cells. Okay? Some are unicellular and some are multicellular. Okay, let's take a little step backwards here. If we're going to consider something to be alive, what are some things it's got to be able to do? What are some characteristics of all living things?
Feed.
They must consume energy of some kind. Yeah.
Reproduce.
Reproduce. Yeah.
Breathe.
Okay. Yeah. Exchange gases in some way, shape, or form. Yeah. Because I mean, plants are alive. They don't necessarily breathe, but they still exchange gases for sure.
That's when you excrete waste.
They have to excrete wastes. Okay. Those are all good. Any others? Okay, so then fire is alive. Fire consumes energy. Fire exchanges gases. Fire can produce waste, and it can reproduce. You get like, a flanker or an ember, right, that jumps out of a fire, it can start another fire. Okay, fire can reproduce. Fire alive?
No.
I mean, it's obviously not a living thing, that it can behave like it's alive sometimes. If you ever ask one of the forest fire jumpers, the guys that jump out of the helicopters and planes and try and fight the fires on the ground, they'll tell you 100% that fire is alive, okay? That it behaves like it has a mind. But it's not, because it's not made up of cells, okay? That is the quintessential characteristic of a living organism. They are made of cells. Now, that creates a bit of a conundrum for one thing, one type of organism. What about viruses? Are they alive? Okay, so you guys just told me that the characteristics of a living thing are consume food, produce waste, okay, exchange gases, be made of cells, okay, and reproduce. Guess what viruses don't do? All of those things. Okay? A virus does not have cells. A virus is a protein shell surrounding a piece of genetic material, either DNA or RNA. Because they have no cellular components, they also have no ability to metabolize food. So they cannot carry out any form of metabolism, burning sugars for energy or anything like that. If they don't do that, they also don't produce any waste. Okay? They don't exchange any gases, okay? And they actually can't reproduce on their own. How does a virus reproduce? Okay, we're gonna take a triggering step backwards to COVID. Okay, we're gonna talk about how viruses work a little bit here. Okay, in the context of a virus is not a living organism. So what happens with a virus is it will latch on to a host cell. Okay, so if I've got, okay, a cell like this, okay, inside the middle of the cell is the nucleus, and that's where all of your DNA is kept. Okay, so all of your genetic material is in there. Right, so what a virus does is it will like latch on, okay, to the outside of the cell. And what it will do is its genetic material is in here. Okay? And what it'll do is it will inject that genetic material into the cell. Your cell is very sensitive about having genetic material floating around inside the cell because genetic material is fragile. Okay? We don't want it floating around inside a cell where it could be damaged. So it detects that now this little piece of viral DNA is floating around and that it might not be safe. So what it does is it will bring it into the nucleus. And that viral DNA will cut your DNA and splice itself in. Okay? Now, here's how your DNA works. Probably learned this in grade 9, but you may have forgotten it. Okay? Every section of your DNA is a code for making something that your body needs. Okay? So there's a part of your DNA that codes for making insulin. Okay? There's a section of your DNA that's a set of instructions for your body on how to make insulin. Right? So let's say that the virus inserts its DNA and cuts out the part that's for insulin. Okay? Is insulin something your body has to make on a regular basis? Okay. So the cell that the virus has infected sends a signal that says, hey, I need the instructions for making insulin. So what the cell does is it makes a copy of the section of the DNA that's for insulin, because it doesn't want to send out the original, it might get damaged, so it just makes a copy. But what's there now? The virus's DNA. So instead of sending out instructions on how to make insulin, it just sent out instructions on how to make a virus. Okay, so those instructions go out. Your cell's machinery doesn't know what it's building, it just builds it. Builds a virus. Now, that may not seem like a big deal, but your cell is a major Karen. Okay, like we are talking that person who flips out when there's a pickle on their cheeseburger. Okay, so it doesn't get the insulin it ordered, and it wants to talk to the manager. Okay, and it goes back to the nucleus and it's like, hey, where's what I ordered? Okay, and the nucleus is just like, the timid manager who's like, oh, we're sorry, we'll make it again. What? No problem, we'll take care of it. Don't scream, don't holler, don't throw stuff, okay, and it just copies that section again. What's still there? The virus's DNA. So it sends out instructions for what it thinks is insulin, and the cell makes another virus. Well, you can imagine that if the Karen gets 2 cheeseburgers with pickles on them, okay, like they are snapping at this point, okay? So they're screaming at the nucleus, where's my stuff, where's my stuff, where's my stuff? And the nucleus just keeps doing the same thing over and over and over again until the cell fills up with viruses and then pops. All the viruses get out, okay? That might be 500 to 1,000 viruses. The infection rate's not super high, okay? Maybe out of the 500, maybe 1% will be successful in infecting another cell, okay? But every one of that 1% makes 1,000 or 500 more, okay? Can you get sick pretty fast, right? You've all had that experience, right? You go to bed and you're like, oh, I think I have a tickle in my throat, and then you wake up the next day and you have to get better to die, okay? Like it happens fast. Okay, it's an exponential rate of growth. But the virus didn't reproduce. It hijacked your cell and made it do it for it. Okay, so a virus is not technically a living organism because it doesn't do any of it. Fire is closer to being a living organism than a virus is. Okay, the virus doesn't do anything on its own. Okay. It's why I always laugh when I see like the container of the Lysol wipes and it says kills flu viruses in like under so many seconds. Do you know how long a flu virus would live on its own anyway?
Under so many seconds.
Yeah, about the same amount of time. Like it doesn't have any cellular machinery. They break down very, very quickly outside of the body. Even inside of the body, they break down pretty quickly. Okay, so they're not a living organism. They're an infectious particle. Okay? So how do you get so sick then? They don't actually do anything.
They multiply fast.
They multiply fast, but like the way you get sick from a bacteria is different from the way you get sick from a virus, right? Like if you get a bacterial infection, so let's say you get food poisoning, that's usually bacterial. Okay, you feel violently ill, you get a fever, all of that kind of stuff. But that's because the bacteria are cells, and their waste products are toxic. So your body reacts to the toxins that are being produced by the bacteria. Okay? When you get infected by a virus, the virus doesn't produce anything. And your sickness is usually a little bit different. You still get a fever, your body reacts to things, but it's different. Okay? The virus isn't releasing any toxins, so why do you get sick? How does your body fight any invader?
Immunity?
Yeah, your immune system, immunity, okay? There's different kinds of cells, okay, that go out and hunt down foreign invaders, okay? When that invader is a bacteria, it can find that, okay? It's another cell. It's obviously not part of your body, and it'll kill it. Okay, and so it's easy for your body to generally take care of those things. And if you get antibiotics, they can kill bacteria, but they don't do anything to viruses because viruses aren't alive. Okay, so the reason you get so sick and so weak when you have a viral infection is the only way to kill the viruses is to kill the infected cells. So your body destroys itself to kill the infection. Going out and trying to find every single tiny little virus that's floating around in the body would be an impossible task and pointless because unless they infect the cell, they're going to break down and be gone anyway. So your immune system goes around and finds cells that are malfunctioning and making viruses and kills them instead. Okay? That's why you get so depleted and so weak. Okay? In order to get rid of the infection, your body has to destroy its own cells. Okay, so it's really a bad situation, right? The immunity cells come over and they're like, ooh, sorry, you're infected, gotta kill you. Okay, like that's what they're gonna do. Okay, they're gonna destroy the infected cell to prevent it from releasing hundreds to thousands of viruses. Okay.
So what was autoimmune?
So autoimmune disorders are where your own immune system goes after your own cells, but usually cells of a specific type. Okay, so like I have something called sarcoidosis, which is a autoimmune disorder where my own immune system goes after parts of my lungs. And I can get like nodules inside my lungs because of my own immune system is going after my own body. Okay? Other types of autoimmune disorders work similarly. It's just a matter of your immune system gets confused and goes, gee, those cells look funny. Maybe they don't belong here. Okay? And so I was kind of joke about it, but yeah, it wants to, it wants to try and kill me from the inside. Okay, but it's, yeah, all autoimmune basically work that way. It's a misidentification of your own cells as something foreign. Okay, everybody kind of makes sense? Okay, so that was kind of really tangential, but okay, all organisms, all true living organisms have to be made of cells. Okay, so we could have single-celled organisms like the actinopods there on the left, multicellular organisms like the cacti there on the right. Okay, we could have single-celled organisms like the paramecium, okay, which you can find in basically any scummy pond water. Okay, amoebas as well. Okay, we'll look at those actually under the microscope next week. Okay, so that's the first point of the cell theory. All living organisms are made of cells. Okay, second point of the cell theory is the one we probably come back to the most, okay, throughout the unit, and that is, what do cells do? Okay, well, the job of the cells is, come on, to carry out the basic functions of the organism. Okay, all the things we do on a large scale, your cells do on a small scale. Okay, respiration, digestion, okay, excretion, okay, reproduction, all of that stuff, okay, can all be done at a cellular level.
Are these the other cell theories?
These are the other points, yeah, 2 and 3. Okay, and the third point of the cell theory talks about how cells are made. Okay, all cells come from the division of existing cells. There's no cell factory in your body that is pumping out new cells. Okay, they all come from the division of cells that are already present.
So when we're born, we have like a couple of cells and they just like,
Yeah, you read my mind. I was just about to get to that. I'm gonna like make everyone horribly uncomfortable and grossed out probably. Okay, so this is one of the kind of incredible things that just is where life comes from. At the moment you were conceived, okay, sperm and egg met. This is the part where you get a lot. Okay, so sperm and egg meet. Okay, the sperm fertilizes the egg. At that point, a cell, a single cell called a zygote is formed. Okay, in that single cell is all of the genetic material necessary to make you. Okay, it is all there at the moment of conception. Okay, incidentally, that's why in religion class, okay, you have probably learned that the Catholic Church believes abortion to be wrong. Okay, the reason the Catholic Church feels that way is actually scientifically based. At the moment you are conceived, all of the potential for you as a person exists at that very moment. I could take that cell and make copies of it and make an infinite number of you. Okay, so you come from that single cell.
And how's a kid born wrong?
That's, I was actually going to get to that. So I will answer that question in just a minute, Harper. Okay. So that single cell divides over and over and over again. It goes from 1 to 2, and 2 to 4, 4 to 8, 8 to 16, 16 to 32, and so on. Okay. It just keeps dividing, dividing, dividing. Eventually, it'll get to the point where you have just kind of a big ball of identical cells. Okay. At this point, okay, at least for like vertebrates, okay, so animals that have a backbone, the cell will fold in on itself, okay, and it'll make a tube inside. Okay, so now instead of being just a ball, the ball kind of pinches in and forms a tube. That tube is what becomes your nervous system, your dorsal hollow nerve cord, your spinal cord, and your brain. Okay, and at that point, the cells start to specialize. Okay, this is the point where stem cells exist. Okay, how many people have heard of stem cell research before? Okay, so in embryonic stem cell research, this is the point where cells are harvested. Okay, so they would take the embryo and they would harvest the cells that are destined to become the liver. Okay, and they put those in a medium and they can grow liver tissue from those cells. Okay, they can do similar things with heart tissue, okay, muscle tissue, all of that. They just take the parts of the embryo, okay, and they can grow them. Of course, what does that mean for the embryo? If I take the cells that were going to be its liver, it's going to die. Okay, that is why in North America, embryonic stem cell research is not allowed. Okay, it's basically saying you're trading one life for another. Okay, because you're going to fertilize this egg. That could be a person. Let's take its liver. Okay, without its consent or anything like that. So that's kind of the reasoning on that. Other countries, they allow it. Okay, like North America does not. So. As you grow as an embryo, cells just keep dividing. Okay? You go from being a single cell to being billions of cells over the course of about 9 months. Okay? So every cell that's in your body now came from that original zygote. Okay? All cells that you make now, when more cells divide, they're coming from other cells. They're not being produced and shipped to the site they're needed. They're the cells that are there and dividing. Okay, now Harvard, to answer your question, how are babies born that maybe have like a birth defect of some kind or have a congenital disease or something like that? That was your question? Okay, so let's use one that's, probably everyone has met someone that has Down syndrome. Okay, Down syndrome is the result of having 3 copies of chromosome 21. Okay, we have 23 chromosomes, 23 from mom, 23 from dad. We have 46 in total. Okay, someone who has Down syndrome, during the initial division of cells, a mistake is made in the copying of the genetic material, and instead of getting 2 copies of chromosome 21, they get 3. Okay, it's not fatal, but it obviously has some pretty major developmental repercussions for that person. Right? They would have Down syndrome with all the physical and mental kind of limitations that would go along with that. But that's the result of the process of division not going right. Okay, and so initially that first cell wasn't right. Okay, and then every cell from there is also not right. Okay, so it happens, it's usually there's all the one or the other of the gametes having a genetic error in it. Okay, diabetes is the same thing. Okay, so if you have diabetes and your body can't make insulin, it's because there's a part of your DNA that has a mistake in it, and that's the part that makes insulin. Okay, your body, when it says, hey, I need insulin, makes something but doesn't work. Okay, it's like insulin but doesn't work. Okay,
What about like allergies?
what's that?
Like allergies, like to like certain medicines and like food and stuff?
I mean, that's more of your immune system. There's some research that says that can be genetic, but I couldn't tell you much more than that. I mean, my wife has allergies, my kids don't. Is that because that was a recessive gene and I had the dominant one? I don't know. So yeah, allergies are kind of, and allergies can come on as you age as well. So they could be environmental as well. So yeah, if someone's going to have some sort of genetic disorder, it is usually right at the beginning that it happens. Okay, and that happens fairly often. Okay, most of the things that are a genetic disorder are so severe that the embryo dies before it even has a chance to get to a certain stage of development. Okay, some of them so early that the woman would likely never even have known she was pregnant. Okay, like that the repercussions of that genetic error are so bad that the embryo dies before it's even a few hundred cells. Okay, and some of them, they can come to full term and live, you know, a normal life. Okay, I mean, if you're diabetic, you're gonna have a pretty normal life. Okay, you have Down syndrome, I mean, you're gonna have some limitations, but you're gonna have a life too, right? So some of those things are, you know, a bigger deal than others, we'll say. Okay, so if the cell is going to divide, the first thing it has to do, and this is the most important, biggest job, is copy all of its DNA. Because both of the daughter cells, that's what we call them, daughter cells, okay, the cells that come from the division, have to have all of the DNA. If they're missing any, there's something they won't be able to make and they'll die. Okay, so the first job is copy all the DNA. Make a mistake in that, cells are gonna die. Okay, if that's one of the gametes, as you were asking, Harvey, that means that probably the embryo that's produced from that won't make it out. Okay? All right. Any other questions there? Don't be afraid to ask, guys. If you got questions, I'll do my best to answer. Sometimes I don't always know the answer. That'll make something up. Okay, so those are the 3 points of the cell theory. Okay, like I said, the second one is probably the most important one because we're going to talk about what cells do a lot, and we're going to relate it back to that quite a bit. Okay, so development of the cell theory. Okay, we'll talk about 4 important people. Okay, so I'll let you copy that down there about Hooke, and then we'll talk about Hooke. The doctor, not the captain, by the way. Okay, so Hooke, believe it or not a biologist. He was a physicist. Okay, Hooke invented the first microscope. He was doing an experiment about lens systems and the path of light as it would go through multiple lenses. Okay, that's a physics experiment. Okay, Hooke is also known for Hooke's law, okay, which governs the behavior of springs and elastic objects. He's a physicist. Okay, so he put together these lenses and he found that as light went through these lenses, it would be diverged. Okay, that means the light rays would get further apart, which, when they do that, results in magnification. Okay, so essentially, what happened was this. So he had like a light source, different color here. Okay, he had a light source like this. Okay, and then he had a specimen like this. Okay, and then he had his lenses or lens. Okay, so the lens would have been like a curved piece of glass like this. Okay, so light came from the light source and it went through the specimen. Like so. When it hits the lens, lenses alter the path of light. Okay, and what happened was the light rays that were going from this side were bent and they ended up over here. Okay, this light ray, okay, was, you know, pretty close to center, so it didn't bend very much. Okay, this one close to center didn't bend very much, and this one, okay, pretty far off of center, ended up over here. Okay, so what ends up happening is that we now have a specimen that is greatly enlarged and flip. Because the light that came through here ends up over there. Okay, so the specimen ends up bigger because the light is diverged. This is how a microscope works. Okay, everybody kind of all right with that? So that was Hooke's experiment. He wanted to see what would happen to the path of the light as it went through the lens. Now, obviously he needed a specimen. He had to have something to look at through the microscope. Okay, what he looked at was a slice of cork. Okay, so he took cork from a wine bottle or something, I don't know if he was a drinker at work or what, but, okay, he had a cork there, okay? And he sliced the cork really, really thin and put it under the microscope. This is what he saw. Now, where does cork come from? Wood, yeah, cork trees. Okay, so what he was looking at were dead cells. Now, of course, he didn't necessarily know that's what he was looking at, okay? But what he saw there were a bunch of plant cells that were dead. So all he could see were the walls, which made them look like empty little rooms, like the cells in a prison. That's why he named them what he named them. He thought they looked like empty little rooms or cells. Okay? So his experiment was a success. Okay? He saw cells and then he went, here you go, biology. You're welcome, sincerely, physics. Okay? Here's the most important tool you'll ever have. Okay? I'm a physics guy, so I brag on physics whenever I can. Okay? So Hooke is the first person to actually view cells and made the first microscope. Okay, so he discovered cells in 1665, okay, in a slice of cork, okay. Now his microscope didn't magnify very much, 30 times, okay. Our weakest lens on our microscopes is 40, okay, and we just use that for finding stuff and then we move up to higher magnifications after. Okay, questions on Hooke? Okay, so Hooke saw empty little rooms, okay, he discovered cells and made the first microscope. Now, around the same time, Anton von Leeuwenhoek, okay, had built a different kind of microscope. Thankfully, it did not catch on, okay, it was not easy to use like Hooke's was. But it was able to magnify quite a bit more than Hooke's was. And Leeuwenhoek used it a lot, and he looked at lots of different things under the microscope. And so where Hooke was the first person to view cells, Leeuwenhoek was the first to view living cells. Okay? He discovered cells are not empty little rooms. They're active things. They have lots of stuff inside. Okay? He would have seen cells that move. Okay, so he would have seen amoebas and paramecium. He pricked his finger and looked at the blood under the microscope. Okay, he got sperm cells from bulls. I don't know how. He was brave / crazy. I don't know. It seems like a dangerous thing to try to get. Okay, bulls are not necessarily super cooperative, and they're very big and they have horns and stuff. Okay. So he was able to view lots of living cells. Okay, so he discovered quite a bit about what cells are and quite a bit about what they do as well. Okay, the last two guys go together. Okay, they worked together and they're the ones who are credited with developing the cell theory. Okay, and that is these two Swedish scientists. Okay. Matthias Schleiden and Theodor Schwann. That's how you pronounce it. Okay.
Do we need to know the dates?
No, I'm not concerned at all with the dates. Okay, so Schleiden and Schwann came up with the original cell theory, which, as all theories do, started as a hypothesis. Okay? So their hypothesis went something like this. If cells are the basic unit of living organisms, and tissue samples from a wide range of living organisms are viewed under a microscope, then it should be possible to observe cells in all of the tissue samples. Okay, pretty straightforward, okay, hypothesis that then became the first point of the cell theory. All living organisms are made of cells, because over and over and over again, they were able to confirm their hypothesis. Every sample they took from every type of living organism, animals, plants, fungi, bacteria, okay, all able to find cells, or whenever it comes back. Okay, so that was their contribution. And of course, in observing all of these cells, they also observed what they do. Okay, they observed that different kinds of cells actually do different jobs. And that's where the second point came in. Cells carry out the basic functions. Okay, and they also observed cells dividing. Okay, observed and like recorded that process. Okay, observed that there were different steps to that process, and never ever found a cell to be produced by any method other than the division of another cell. Okay. Now, their equipment, much better. Still a light microscope, but up to 600 times. Our microscopes max out at 400. Okay, but that's still sufficient to see everything we need to see. Okay. Okay.
If, would scientists like need the 600 times magnification right now?
They would, because they'd be looking for more specific parts of the cell. Like, we could put another lens, like there's another spot for a lens on all of our microscopes. It's called an oil immersion lens. They require a lot of extra maintenance, like cleaning, and you have to have the oil around all the time, so they're difficult to use, so we don't bother because we don't need to see that much detail. You could see, instead of just seeing the cells and some of the bigger parts, you'd be able to see some of the smaller parts. Okay, let's take a couple minutes here, just get up, walk around a little bit, and then we'll continue on. Yes. So in this diagram here, we got plant cell on the left, got animal cell on the right. Okay? Do they have a lot in common? Okay? Yeah. So they have a lot in common, okay? More so than they have different. Okay, like, I mean, the differences are obvious because that's kind of the way our brain is wired. Our brain is wired to notice differences before it's wired to notice similarities. Okay, it's what allows us to tell like faces apart and things like that. It's just the way our brain is kind of evolved and wired that way. Okay, so when we look at this, we go, oh, geez, those are not at all alike. But if we look closer, okay, we can see that they actually share quite a bit of things. I mean, every single well, I shouldn't say every single. There's only a few of these that aren't shared by both, right? They both have a nucleus. They both have nuclear pores, nuclear membranes, nucleolus. They both have chromatin. That's DNA. They both have mitochondria. Okay. I would say that the vacuole one, they're different kinds, so that would be one that's probably not the same in between. They both have ribosomes. Okay, here. Okay, and this one has ribosomes there. Okay, lysosomes, that would be another one that's only in animals. Plants don't need that one because it digests food. Okay, they have Golgi apparatus, both of them. Both have smooth ER, rough ER. Okay, they both have cytoplasm. They both have cell membranes. Okay, like there's a lot that they have that's the same.
So like there's those plants somewhere where they're like a bell and the rats fall in.
Mhm.
Would they have the digestive cell?
They will not have the lysosomes. They can make some enzymes. So your carnivorous plants, like your pitcher plant, I think is the one you're describing. Okay, those are not surviving on the energy they get from the things that fall in. They're more getting like minerals and stuff. They grow in soils that are nutrient poor, but their primary method of getting energy is still photosynthesis. Okay, so even like a Venus flytrap, a pitcher plant, whatever, the vast majority of their energy is still coming from the sun, but they will get some nutrients from the things that they eat. Okay, but their digestion is not at all like ours. Okay, it's not nearly as complete. okay? If you're looking at like a pitcher plant, it'll eat only, if it's like something big like that, that's the only, that pitcher will die, okay? Because it's not gonna be able to completely digest all the tissue, the bones, or any of that, right? Whereas if you have an owl, for example, and they eat a mouse, okay, their droppings contain just the bones, right, and the hair and stuff like that, right? So it's chemical digestion, but it's not necessarily for energy's sake. If that makes sense. Okay. So they wouldn't have that organelle, but they would have the ability to make some chemicals. For an animal, the digestion actually occurs inside of that. Whereas for a plant, the digestion would be occurring in the vat, right, in the pitcher. Okay, so the idea then that these have a lot of things in common is what led scientists to start looking along the paleontological history of life on Earth. Okay, and what they've discovered through micropaleontology, so that is paleontology done through a microscope, looking at microscopic organisms, is that all life can be traced back to a single cell ancestor. Okay? Something that was neither plant nor animal and then diverged into plants, animals, fungi, okay? The protists, okay? The monorins, all that kind of stuff. So there's different groups and kingdoms of animals, some of which have gone extinct over the years. Okay? But we know that all life on Earth did actually come from a single cell organism. Okay? That is why there's still a ton of structures in common between a plant cell and an animal cell. Okay? It's way more likely and backed by scientific evidence that they have a common ancestor rather than they evolved completely separately, but look how alike they look. Okay? Think of it this way. There's a reason you look like your mom and dad. You share a lot of genetic material with them. Okay? Is it possible that somewhere out there in the world you might have a doppelganger? But it's not likely, right? Okay? And the reason they look like you isn't because they share any genetic information with you. But that's way less likely than it is for you to look like your brother or sister or your mom and dad. Okay? So these things look alike because they had a common ancestor, not because they magically look like doppelgangers later. Okay? That's way, way less likely. All right? Okay, so generally speaking, okay, the science says that, yeah, everything came from a single-celled organism about, 3.5 billion years ago or so. Okay, we are going to do some microscopy stuff next week, okay? So I am just going to run across the hall and grab a microscope just so I can explain this stuff here, okay? But if you don't have the notes, I want you to copy down what's on the board right now while I grab that microscope. Okay, so one on the arm, one on the base, okay, at all times, and always have control of the... And that's why we carry them with two hands. Somebody obviously wasn't carrying that with two hands. That's why this is all taped up. Okay, this happens all the time. We got to make sure we know what's going on with the cords. Okay? Because if we don't, what happened to this one is clearly what will happen. You'll be walking along and you'll step on that cord and you'll pull the end of it right off or drop the microscope or both. Okay? And then you'll hear the crashing sound. Then you'll hear the angry yelling sound, thumping sound, ambulance sound. That got dark fast, didn't it? Okay, so just be careful with them because obviously now this one is wrecked. Okay, we'll have to bring in Mr. Vanner, the super sub, to fix them. Okay, so I'm never going to ask you to label a diagram of a microscope. Okay, you never have to memorize the parts. But you need to know what they are because those are the terms I'm going to use, in class when I'm talking about a microscope. Okay, so for our microscopes, okay, these ones are good microscopes. They're old, but they're good. So they all have 3 lenses. Okay, the lenses are, okay, the shortest one, this is the scanning power lens. You use it for, oddly enough, scanning a slide to find the specimen. It only magnifies 40 times, so you're not going to see a lot of detail with it, but you'll be able to find the specimen pretty easily with it. Once you find it, then you will move up to the next power, okay? The next power is the one that's medium in length and has a yellow band on it. This is the low power lens, and it magnifies 100 times. Okay, once you find what you're looking for, if you still want to see more detail, then you go up one more, okay, to the high power lens. That's the longest one, it has a blue band on it, okay? And at that point, that's as far as you can go. Okay, everybody with me there? All right, we also have, okay, the revolving nose piece, okay, that's this part here, and then we have the revolving eye piece up here, okay? The reason that this turns is so that you don't have to lift or drag the microscope for your partner to look in it, okay? Because the microscopes are a sensitive piece of equipment. If I pick it up, if I get it all focused, pick it up and hand it to my partner and set it back down again, that knocks it out of focus. Now your partner's got to start all over again. But if you're both sitting at the table, okay, you get it all in focus and then go, hey buddy, have a look, okay? Now it stays in focus, right? Because you haven't had to lift it up and put it back down. Making sense? Because I see people do that. They're like, they look through it and then like, hey, and they turn it around and they say, we can't see anything, you suck, you didn't focus it, and then there's a fight, and, okay? We don't need that, okay? Other parts of this microscope, this is probably the part that's the nicest feature of these, is that the stage, okay, that's the part where the slide goes, can move around using these little knobs. Okay, so these little knobs here, okay, move the stage. So you just put the slide in this thing here, let it close on it, and then you can just move the slide around rather than having to put your fingers on the slide and move it this way. Because what happens usually when you do that is you push the slide down, which moves it out of focus. Okay, these allow you to move it without losing the focus. So it's a nice feature that these ones have. Okay, underneath the stage, okay, is stuff to do with the lights. We probably won't have to adjust much of that. Okay, and then obviously power switch on the back and a rheostat here to increase or decrease the brightness of the light. Okay, everybody good with the parts on that? Okay, so we carry it with two hands. Okay, whenever you're done with a microscope, you always return it with the scanning power lens in place. Two reasons for that. That's what the next person who uses it is going to want. Okay, so it's polite. It's also safer because if you leave the high power lens in place and the person who grabs it assumes the short one is in place, they could damage the lens by cranking on the focus knobs and breaking the slide, like ramming it right into the lens. Okay. Something I forgot to mention. Two different focus knobs. Okay, this one is the coarse focus knob. Watch how much the stage moves. Okay, like you can't even make a full turn with this knob before it's gone through the full range of travel. So this one focuses fast. Okay, the fine focus knob is geared differently. Okay, you can just crank away on this thing. Okay, and it barely moves the slide at all, okay, so you can do really, really fine focusing. You'll use this one when you're on the high power lens for sure, okay? You'll never use this one on the high power lens. Okay, when using a microscope, always start with the scanning power, lowest power, and move up from there. Okay, and only use lens paper to clean the lenses. From time to time, the lenses get dirty. Okay, people have oily skin. Okay, especially if you're wearing mascara is the stuff that goes in your eyelashes. Right? Okay, that stuff that's really oily. Okay, and when it falls on the lens, it can really make a mess of it. So maybe on lab day, don't do the lashes that day. But if you have to, okay, you might put grease. Because that stuff's greasy. Okay, on the lens you might have to clean it. Okay, there's special paper for that. I will have it at the front. Please, for the love of God, don't do this. Okay, that's how people get pink eye. That's gross. Okay, no, not really. You shouldn't be hitting your eye on this ever, but that's gross. Okay, also not this. Okay, you could have grit on your thumb, fingers, okay, and that grit could be microscopic, you don't know it's there, but you make a microscopic scratch in something that magnifies stuff, that scratch goes looking like the Grand Canyon when you look through the microscope, okay? So you always use this lens paper because we know it's clean, and once you use it, you throw it out, you don't use it again. It'll pick stuff up, and we don't want it to be used on the slide, on the microscope. Okay, questions on that part? So just careful handling so that we preserve the equipment. Okay, parts of the microscope, we already did that. Okay, here's what I need you to do. I need you to copy down what's on the slide right now, or on the screen right now, and then I will explain it. Okay, so for right now, just draw this diagram, okay? And I know this question will come up. What is this symbol right here? Okay, this symbol is mu. It's the Greek letter M. Okay, and we use it for micro. So this is 4,000 micrometers. We don't want to use M because M means millimeters, and those are different. Okay, so it's just a U with little tails. Here's what this diagram is trying to relate to us. It's comparing what we call the field of view, so the size of the area you see through the different lenses of the microscope. Okay, so when I look through the scanning power lens, okay, let's say I see the specimen that I'm looking for, okay, and let's say that that specimen is about yay big when I look through the scanning power lens. The scanning power lens doesn't magnify very much, so things aren't going to look a whole lot bigger. Okay, so it's not going to take up very much of the area that I can see. Okay, the purpose of the scanning power lens is to find things. Right? So if I move up to the next magnification, okay, will that object now look bigger? Obviously, I've got a higher magnification, right? Does it also take up more of the area that I can see? It does. And then as I move up even more to high power, right, it probably takes up the entire area that I can see. So as we move up in magnification, yeah, things look bigger because we're seeing less and less space through the microscope. The circle we see through the microscope looks the same, but it represents less space. Okay? When I look through the scanning power lens, the diameter of the circle that I see is 4,000 micrometers. That's 4 millimeters. Okay? When I move up to the low power, I go from 40 times to 100 times, and now I shrink the area that I see. It goes from being 4,000 micrometers to only 1,600 micrometers. But obviously the thing I look at takes up more of that space. When I move up to high power, I'm increasing the magnification, okay, from 100 to 400, okay, and now that thing takes up basically the entire field of view. Okay, everybody all right with that idea? Okay, so the microscope kind of works like this. Let's say you go to a Flames game, okay, so you're in the Saddle Dome, okay, and you get a text and it's your buddy. And they say, hey, I'm here too, okay, where are you? Okay, but you're not, you're like me and you don't know where the seats are and where the sections are and whatever, so you're like, I don't know, but I'm wearing a bright yellow shirt. Okay, I mean, everybody else is going to be wearing a Flames jersey. You're the one wearing the bright yellow shirt. Okay, so if that person wants to find you, are they going to grab a high-powered telescope and scan every single person in the crowd looking for a yellow shirt? No, they're just going to use the naked eye and go around until they see something yellow, and then maybe they'll put their binoculars on and go, oh yeah, that is them. That's you over there, I see you. Okay, but they wouldn't initially go to the highest magnification to find their friend because it would take too long. They want to scan a wider area, okay, first, and then when they think they found you, zoom in on that. That's what you're doing with the microscope. Scan a large area to find it, okay, get it in the middle, and then magnify it from there. Okay, making sense on that? Because I see people do this all the time. I give them a slide, they go straight to high power, and they can't find anything. Yeah, you can't, you won't, okay? You're trying to find your friend with the yellow shirt with a high-powered telescope by scanning every single person of the 18,000 people that are there, okay? That is not going to happen, okay? Start on scanning, find it, and then move up, move up. Okay, the reason we need these numbers is we need to be able to estimate how big what we're looking at through the microscope is. All right, so if I take my little specimen here, okay, and I bring it back over here, if I know this distance is 1,600 micrometers, could I estimate how big that object is? Right? It's just an estimate. I'm not asking you to calculate anything or like physically measure something. Okay? If I look at that, I think I could probably fit, what? Let's say 10. I could probably fit 10 of those from one side to the other. Okay? So if I can fit 10 across and it's 1,600 across, how big is it? 160. How'd you do that? In your head. You went 1,600 divided by 10? Okay, got 160. All right, that's all we have to do for estimating the size of an object. Just estimate about how much of it would go, how many would go across, divided by that number, you've got the size. Okay, and it doesn't need to be any more accurate than that. It is, after all, just an estimate. Okay? Everybody all right with that idea? All right, so make sure you have that diagram, because when we're doing lab diagrams, which we'll talk about next week, okay, that's going to be one of the things you'll be asked to do, is estimate the size and put it on your diagram. Okay, I would like you guys to answer those 3 questions, okay, on a sheet of paper, and we'll go through them here before the bell goes. Cell theory. Okay, 3 points of the cell theory are: all organisms are made of cells. Okay, cells carry out the basic functions. Cells come from the division of existing cells. Okay, 4 people considered most important: Hooke developed the microscope, discovered cells. Leeuwenhoek discovered cells were not empty and looked at living cells. Okay, Schleiden and Schwann found cells in all living things and developed the cell theory. Okay, that's who you need to know. All right, and number 3, on what basis can we deduce that plants and animals may have had a common ancestor? They have a lot of similar structures. That's what we learned today. Later in the unit, we'll learn they have a lot of similar processes too. Okay, test tomorrow. This is not on it. What we learned today is not on the test tomorrow. Don't study this, okay? Just the chemistry stuff. Pencils, calculator with fresh batteries, periodic table with special elements, charges, and prefixes on it. Okay? That is what you may have written on your periodic table.